Last week, Dr. Bronowski described the events that culminated in the dropping of an atomic bomb at the end of the Second World War. This week, he indicates how those events led to many physicists turning their attentions to biology in an attempt to unravel the structure of life. Dr. Bronowski himself changed from the physical sciences to biology when he moved to the Salk Institute at San Diego, California. He became the director of the Council for Biology in Human Affairs.
We have spoken much of physics and the 30s, but any 20th century man's skull is deeply engraved with the transition between the 30s and the 50s, with a war in between. Human life was threatened, lost. The atom bomb was invented. Fermi, I think, said, "There goes the end of a beautiful subject." To me, the 50s are the exciting time in science because of a great explosion of biological discoveries.
That's right. That prophetic remark, "There goes the end of a beautiful subject," namely theoretical physics, does say something about 1945. Physics ceased to be a dream because it had struck against harsh reality. It also ceased to be so inviting because wonderful new ideas were coming up in biology. And those things are not independent. And I'd like to start by saying something about the biographical history of that time. Between 1939 and 1945, every scientist in the West was working in some way on war projects, winning the war, defeating Hitler, preserving the tradition of culture and science were the great aims of 1939 to 1945. And a whole generation of scientists who were bursting into the world with promise in, say, 1940, by 1945 were asking themselves, what do we do next? I'll take my own case. In 1939, I was a young man of just over 30, just on the threshold of what should have been my most productive years as a mathematician. By 1945, I was 37. The big papers ought to have been published. And I did publish some big papers, but they were all hidden in secret archives, in war offices. My younger colleagues felt this even more, and a very strange pilgrimage took place. Everybody left the army in 1945 and 1946. Everybody had their gratuities. And everybody, instead of going back to the physics lab, said, just a moment. When I left school or stopped being an undergraduate, why, then I was certain that I wanted to be a physicist. But now I've been a physicist for 6 years. And once I walk through those laboratory doors again, they will shut on me forever. Isn't this the moment to think of whether my career should really be in physics? And many people decided otherwise. Some decided for personal reasons. For instance, I know some people who were simply fed up with being followed about by the Secret Service wherever they went, because they were in high, what were called sensitive projects in physics. Others, like Leo Szilard and some of my older colleagues, wanted to get away from physics because they had been very much against the dropping of the bomb. Szilard, whom I knew very well, who was my colleague in the last year of his life at the Salk Institute, and who in many ways was the inspiration of the Salk Institute, was actually instrumental in bringing me to the Salk Institute because I was the first scientist who talked about human values. And I talked about human values because I went to Nagasaki and Hiroshima in 1945. I saw what the bombs had done, and I was so aghast that I decided that it was not only necessary not to do such work, but to spend the rest of one's life speaking out against it. How was the Salk Institute founded? By people like Szilard and me, who said, we've done all the physics we're ever going to do. We're now going to do biology. We're going to do a subject which does something to bring to the idea of life the same sense of devotion and passionate intensity which we have brought to the understanding of the physical world. But when we brought it to the understanding of the physical world, we were acting with that loyalty to human values which the bomb itself carried. And finally, other bright young men, take Francis Crick, for instance, one of the leading figures of the new biology, had worked on making and laying mines during the war and now wanted to do something new, and they had the chance. So much for the human element. Now the scientific element. In the 1930s, people had begun to talk about the secret of life. What makes a living thing different from a dead thing? In the 1940s, they began to suspect that the secret of life was not as complicated as biologists thought. It's a very nice idea.
It always seemed to me that the ideas of science are so simple that you have to be quite intelligent to understand them.
That's a very shrewd and complimentary remark. Yes, we scientists are rather simple-minded. We say if it's hard, it's probably wrong. Good ideas are very simple. Biologists, on the whole, had thought that life must be written in the proteins, that is, in those very big molecules containing many thousands of atoms, in which the actual bodily exchanges take place. We take in proteins in order to live, we break them apart, we reconstitute them, we then throw out some of the material, and that exchange makes life. Now, the disadvantage of the proteins is that they're very complicated. At least they're very big. They may turn out not to be as complicated because they may turn out to be assembled from simpler units which at the moment we don't understand.
And it may be a simple rule which says why they coil and twist in what look like complicated.
Biggest problem in biology at the moment, if I were 40 years younger, I would set myself the task of why is the configuration, the shape of the proteins, determined by the actual way these atoms are aligned together. And since we don't even know that yet, and we are now in the 1970s, we are, 30 years after the event. In 1940, working with the proteins was certainly going to be very hard. And then, during the war, a man called Avery began to produce evidence that actually the secret of life was written on a much simpler chemical compound, a nucleic acid. Now, anything that's an acid in the nature of things is much simpler than a protein. And a nucleic acid is an acid in the nucleus of each human, animal, or vegetable cell. And Avery was the first man who gave rather good evidence, which I won't detail because it would carry us too far away, that the nucleic acids played a bigger part, and in particular, one of the main types of nucleic acid, deoxyribonucleic acid, usually called DNA. The Na is the important part, N for nucleic, A for acid. The D standing for deoxyribo just specifies the kind, but the important thing is it's a nucleic acid. It's an acid, that means a fairly small molecule. And it's a nucleic, that means it occurs mainly in the nucleus, the central content of cells. So, after 1945 to 1950, people began to take a healthy interest in nucleic acids. And they discovered all kinds of interesting information. It was pointed out, for instance, that the nucleic acids are made up of sugars and phosphates for structural reasons, and otherwise of 4 main kinds of really rather small molecules, none of them containing more than 12 atoms. And that these 4 molecules, as it were the letters of the alphabet, shuffled about in different ways, made up the nucleic acid. And there were pairing rules as to how much of one would go with another, all of which were there for everybody to see, that God had scattered the ground as thick with clues as autumn leaves, but we missed them all and kept on stepping in the puddles. In 1951, a rather bright, rather brash, rather ordinary young American, whose chief distinction until then had been bird watching, got himself a scholarship to Europe to study nucleic acids. And he went to Scandinavia. Like most Americans, he spoke foreign languages execrably. And he made no progress at all in Scandinavia. For one thing, he couldn't ride a bicycle very well, which turned out to be an essential part of the laboratory ritual. For another thing, he couldn't make himself understood by the professor he was supposed to be working for. And third, and most important, he actually never understood the word the professor said, but he didn't like to tell that to the professor. So in an agony of distress, he was a very shy young man, he decided to go to a country in which they spoke his language. In 1951, he came to England, to Cambridge, and there he teamed up with a much older man, and they together attacked the problem of the nucleic acids. The young man was called James Watson. The older man was called Francis Crick. They made a strange team because Watson was only in his 20s, but Crick was in his 30s and should have had his PhD, but had been delayed by the war. Between them, in an incredibly short time, they unraveled the structure of this nucleic acid, namely DNA. And they published the structure in the spring of 1953, when other people had hardly yet woken up to the fact that they were working on it.
I remember the paper very well. It really came out like a bomb.
Yes, it was well written, it was well publicized. Now, their structure of DNA had several very attractive features. First of all, it was simple. The information was carried by 4 letters, as if it were printed. These 4 smallish molecules: adenine, cytosine, guanine, thymine. Secondly, the information was carried on 2 parallel strands, and what was written on one strand was always matched on what was written on the opposite one. So the information was duplicated.
But in reverse, it paired.
They were paired.
Every head information had a tail on the other side.
Right. But thirdly, this lent itself particularly to understanding how it would work when a cell divided, because it then became clear that the 2 strands would take apart. One would go into each half of the cell to make a beginning for a new cell, and would there form on itself a parallel match. So we had a duplicating process, a little like a chain reaction. Well, it was a beautiful time, because it suddenly showed that biological phenomena could be described in very simple terms, that there were very exact physical and chemical relations which determined it. It showed that the language of life was universal, in that every cell was given its special properties by exactly this same thing, a particular DNA that said, this is an elephant, or this is a rose, or this is a banana.
In fact, it gave a rationale for the working of Mendel's genetics.
Laws of heredity. Indeed it did, by this very process of division, by showing that the information that was being carried in what had always been recognized as the chromosomes by the genes was in fact DNA information, nucleic acid information. But you see, it showed the universality of life, but it also showed something which I can't stress enough. The special character of life is due to the architecture of the molecules, not to their kind. For thousands of years, men had been quarreling about the spirit of life. What makes a living thing? What makes it special? And it had always had to do with a spirit is breathed into you. A soul arrives at the moment of your birth and leaves at the moment of your death. Your body is animated by an intangible quality. The word spirit entered into all these descriptions. And here at one blow, we stepped out of this vague, inexact world into the exact world which had been created by the physicists, a world of atoms. What was said by Francis Crick and James Watson once for all is, all life has a special character, all life has the same special character. It is made up of quite ordinary atoms, but it's alive because the atoms are arranged in an architecture which lends itself to reduplication and therefore to the carrying on of life. So that's the definition of life. When is a thing alive? When every moment of its existence repeats a process which ultimately will be handed on from it to its offspring. And the architecture of DNA is a physical structure which shows just how this happens, both when a cell divides and when cells unite to form a future creature. I said earlier in these talks that I did a kind of mathematics which was Greek. Plato said, not God is a mathematician, that's a 20th century saying. He said God is a geometer. He is the kind of mathematician who deals with forms. I've been that kind of mathematician all my life, and that's why I took to biology like a fish to water.
That was the late Dr. Jacob Bronowski in the 7th of 10 conversations he recorded with George Steedman. Next week, Dr. Bronowski discovers that evolution is progress and suggests that evolution favors more complex structures because complexity is more stable with greater potential for adaptation. Each of the talks in this series is being printed in The Listener, and Dr. Bronowski's book, The Ascent of Man, is available from most bookshops, price £5.50.